Hi everybody, I start through lecture and then continue from amino acid. Today I want to speak about the structure and function of the amino acid. Amino acids are important organic compounds composed of the amine group, carboxylic acid group, hydrogen and side chain R group which bind to the alpha carbon. This carbon his name is the alpha carbon, and because of this we call alpha amino acid. Other things because of NH2 group in the left side we call L amino acid. As I see in this picture also I can show here amine group in the left, R group, side chain, hydrogen, alpha carbon in the center, and the carboxylic acid group. Now we classify the amino acids. Amino acids can be classified mainly in two groups, polar and non-polar. First group is non-polar amino acid, is aliphatic. Usually the side chain is hydrogen and carbon, and in the exception methionine is S, sulfur which is between the two carbon. In here, all of the amino acids are chiral except glycine because we have hydrogen and two hydrogens alpha carbon. Glycine is the side chain is hydrogen, alanine side chain is the methyl, valine side chain is the isopropyl, leucine side chain is the isobutyl, methionine we have a methyl ethyl sulfide, and isoleucine which is secondary butyl is which bind here. Okay, as you see here all of these are aliphatic R group, it's not cyclic, and also as you see here they are non-polar. Okay, usually they can solve in the non-polar solvent. The second group, again, they are non-polar. Okay, non-polar. And it is aromatic group. For example, phenylalanine. Okay, tyrosine has the high polar compared to the phenylalanine. And tryptophan. Okay. And the third group is the polar uncharged R group. Okay, which can have oxygen or nitrogen or sulfur SH in the group. As you see here in the serine we have a methylene hydroxide, threonine we have a ethylene hydroxide, and here is the cysteine we have a methylene sulfide, thiolmethyl, excuse me, and proline only amino acid is the secondary amine. And also we have asparagine is amide group, glutamine is amide group. Okay, or they are in the polar uncharged R group. And also we have again fourth group. Again they are polar positively charged R group, which is we can call it also basic because they have amine group is like lysine. It is arginine, histidine, okay, and you see they have a amine group, and the last group is the fifth is the polar negatively charged R group is acidic, which one is aspartate. It has acid carboxylic acid here, and glutamate also here. The acidity of the aspartate is more than glutamate because of the less amount of the methylene group. Okay, amino acids are building block of protein. Protein are copolymer of the 20 different amino acid. Post-synthesis modification of protein results in very high number of amino acid derivatives. It's very important. We can add the sugar, acetyl group, phosphate group, and we can make more modification of the structure. And the amino acid, and also the amino acid found in the protein are alpha amino acid, which is important. Okay, all of them are primary except proline, which is secondary. The side chain of amino acid can be polar, which, as I mentioned, this can be soluble in water. Non-polar can be soluble in non-polar solvent. Polar amino acids are either neutral or negatively or positively charged, acidic or basic, which I mentioned, and the side chain can be aliphatic or aromatic. And also you can learn about the essential amino acids, very important. I can show here, for example, with the green color. For example, as I know, leucine is essential, okay, and the lysine is essential, okay, and histidine is essential, isoleucine is essential, valine is essential, tryptophan is essential, and also threonine is essential, and phenylalanine is essential, and histidine also essential, and it must be 9: leucine, isoleucine, valine, okay, lysine, histidine, and also we have to find the methionine is essential. These are essential amino acids which body cannot produce, okay? Other thing is very important, I have to explain it. We can have a two type of structure, okay? One type we can write in form, for example, I draw the glycine, okay, as a normal structure, but sometimes we draw in the Zweitter ion form, okay, which carboxylic acid lose hydrogen and become negative, and the proton and the amine group accept hydrogen and become positive group. As you see here, it is a positive, it is a negative. We can use a Zweitter form, which I explained about the structure in the chemistry lecture. How the protein form, okay? As you know, for the formation of protein, we need to first form the peptide bond, okay? But also the side chain may have interaction like van der Waals force, hydrogen, complex bonding, ionic, and or salt bridge, covalent bond, or disulfur bridge. Combination of 20 amino acid result amazing variety of the polypeptide and protein. For example, we have a 3 to 2 million pentapeptide. Okay, it's very, very big. Also, amino acid, 20 amino acids used in synthesis of protein. Active protein contain more than 50 different modified amino acids. Can be phosphorylate, acyl group, glycosyl group, hydroxyl group, and the methyl group. Okay, for example, methyl group is the CH3, OH for hydroxyl, glycosyl is the glucose, acyl is the RC double bond O, and the phosphate group is the PO43 minus. Okay, acid is CH3CO, and sulfur is the S, and etc. First up, we can classify the structure of a protein from the formation. Is the first one is the primary structure. It is a sequence of amino acids in the primary, which the backbone of protein is the monotonic chain with repetition of NCC, and also they bind the amino acid together by the peptide bond. For example, if you see here, the first amino acid is here, okay. Second amino acid is here, okay. The first one loses OH group, the second one loses hydrogen, both of them gives water, and the carbonyl group bind the amine and formation of peptide bond. And then this bond, okay, is called peptide bond is here, okay. For example, as you see here again, if we have form of Zwitter shape again, the same. In this case, it 2 hydrogen from this group, 1 oxygen from this group, again peptide bind form, and it is a dipeptide. We can continue to 3 peptide, 4 peptide, tetrapeptide, pentide, and the biggest primary. Okay, if you see the biggest primary, start with the amine group, we say N terminal, and finish in the C terminal. Then it is good, very important. You know, we have a I choose here the peptide bonds. Okay, M minus 1 peptide bond. It is a 4 peptide bond. Okay. But what is a secondary? For example, the flexibility of the polypeptide chain and rotation and form a different conformation. Many different structure can be formed, and it will be random coil, alpha helix, beta sheet, and the turn and loop. For example, it is alpha helix. Okay, the hydrogen bond usually between carbonyl and amine group. Okay, and NH group, hydrogen of NH, and between the amino acid number 1 and the 4. Okay, it is a helix form, it's very important. The alpha helix is stable, but a very flexible structure, stabilized by hydrogen bridge, hydrogen bond between the NH group, okay, as you see here, and the carbonyl group, excuse me. And as you see here, side chain also of amino acid are on the surface, are helix, okay, determine the polarity of the surface charge of the helix. Okay, if it is a polar, you see it is a side chain. We can say the side is polar. Okay. The structure is stabilized by hydrogen bond can form also between the polypeptide chain running the parallel. This sheet like form called beta sheet. We will see in the next page, as you see here. We have 2 types of beta sheet. If the same direction to the parallel or opposite direction is anti-parallel, and the hydrogen bond between 2 is opposite amino acid between the amine group and the carbonyl group. Okay, and also this is short, as I mentioned. They can be parallel and anti-parallel. Beta sheet is usually short, is between 5 to 8 amino acid long, and also stable, make protein with beta sheet very durable. Okay, sheets are slide over each other. Okay, like elasticity of silk. The beta sheet can run parallel and anti-parallel, which I showed you. Okay, and also we have other structure like terns or loops or super secondary, which we study in the biochemistry. Okay, but we go to the tertiary. Okay, before I start the tertiary, I explain. Okay, I draw one amino acid. Okay, for example, I start from NH2. Okay, and finish with the COOH, and I have a different amino acid. Okay, with primary structure, and as you see here, now I draw the several secondary. Okay, for example, start from NH2. First one I draw alpha helix for the, okay, hydrogen bond between the first and the fourth amino acid. Then there is a loop, 1 hydrogen bond here. Okay, then I start beta sheet, again loop, excuse me, beta sheet, okay, Okay. And again, I will have alpha helix here with the hydrogen bond between beta sheet. Okay, and continue again loop, and again alpha helix. Okay, with the hydrogen bonds here. Okay, this is it. As you end in the end, COH is the finishing here. Right to the secondary, we can show it is alpha helix, loop, and secondary is a beta sheet, turn, and also alpha helix, again loop, again alpha helix, okay? Now it is a secondary, different secondary. When it is secondary, more flexible because these groups, it is freely can move. This freely can move. But maybe, for example, here we have a cysteine. It has a SH group and the other cysteine. It has a SH group. Okay, remove the S hydrogens. Okay, and the S group bind each other. It is called disulfur bridge. For example, between here cysteine and with here cysteine. Okay, as you see here, it causes this alpha chain, alpha helix can bind to this beta sheet. Okay, maybe we have a cation anion here, for example, aspartate. Okay, and we have a, for example, lysine here, group side chain, NH3 plus, and they make a salt bridge or ionic interaction. For example, here again. Okay, maybe we have, but then I write here, so, excuse me, write same color, salt bridge or ionic interaction. Or we can have a disulfide bridge between two cysteine. Maybe we have also group interaction between, for example, I have here valine. Here I have a leucine. Here I have a isoleucine. Okay, between them there is a hydrophobic interaction. Or we can say van der Waals, or okay, nonpolar interaction. As you see here, we have it three main, or we can make a new hydrogen bond, for example. New hydrogen bond. Okay. New hydrogen bond. Then I can write the hydrophobic interaction. And we have also new hydrogen bond. This structure altogether called tertiary structure is more fixed, more stable, and easily can be modified. Okay. What is the tertiary structure? The tertiary structure is the final specific geometric shape of protein. The final shape is determined of the variety of the bonding interaction between side chains of amino acid, as I mentioned. This bonding interaction may be stronger than the hydrogen bond, and between the amide group and in the alpha helix in the helical part. As a result, bond interaction between side chain may cause a number of folds, and the shape can be changed. Bends and loops and all domain of the polypeptide contain tertiary structure. There are four types of interaction, very important: hydrogen bond, salt bridge or ionic interaction, disulfur bridge between two cysteine, and nonpolar hydrophobic interaction between the nonpolar side chains. Different fragments of same chain may between the bond together. Now we understand how is the tertiary structure formed. What is the coordinate structure? The coordinate structure of the protein tell you how many subunits of polypeptide chain are in the protein. For example, hemoglobin. Okay, it's the quaternary peptide, and it has a 2 alpha and 2 beta, which is the 4th subunit. Okay, proteins can be homo, heterodimer, trimer, or tetramer. For example, hemoglobin tetramer. A large number of the protein have a 6 or even more subunit. As you see with the tertiary, if we have several tertiary, we can have a quaternary. As you see here, it is a primary, which has the N-terminal and the C-terminal. Alpha helix, it is between the primary, it is the amino acids and the first and fourth. We will have a tertiary, which include many alpha helix and the beta sheet. And if we have several number of the tertiary, we can find the quaternary. What is the biological function of the protein? Hormones for the regulation of the body, usually produced by the glands, okay, endocrine glands, cytokines, which is important for the immune system, ligand which can bind to receptor. They can be usually proteins, okay. For example, could have intracellular communication, cell community exchange, and also regulation of like hormone, regulator and protective like cytokine. Receptor and cell adhesion proteins, they are very important participation in the intracellular signaling, transduction, and also regulation of gene, and also communication between the cell. And we have a channel for the pump for the passing material from membrane. For example, pump, they are active transport from low concentration to the high concentration. They need the ATP channel for the passing material, for example, ions, okay. Transported like a gate or carrier, okay, we will study later. This protein controls the exchange of the material between cell, environment, and outside of cell. Also, some of them needs energy because like it is active transport, use the ATP or GTP. And thank you very much for this protein and amino acid groups. And next lecture we continue with the nucleic acid structure, and see you next time.